yeast have a two-step process. And so first of all, they release the carbon dioxide from the top of the molecule, and it leaves this little short acetaldehyde down here. And the carbon dioxide, what are yeast good for? Good for baking bread. Okay, and so this is a breakdown product of sugar. So you got the sugar in the bread dough, and you put the yeast in the bread dough, and they start digesting the sugar anaerobically, and they produce carbon dioxide bubbles that makes the bread rise. Okay, well, what else? What do you do with this little molecule? Now you add the hydrogens to this, and what do you get? Ethyl alcohol. So this is what? Vodka, Jim Beam, etc. Okay, and so this is the famous drinking alcohol. Now for bakers, that's no problem because the alcohol all evaporates. Okay, but for brewers, they just let the carbon dioxide bubble off. They keep this part. And I had a rather interesting biochemistry teacher. And one of his basic reasons for rejecting God was that we had lactic dehydrogenase, whereas the yeast had a decarboxylase and a dehydrogenase. And he thought if God really loved us, that he would have given us this enzyme. So when we got tired or frustrated, we could just tie a plastic bag over our head and just breathe a few times, and we'd be drunk as a skunk and forget our worries. Fortunately, God had a much better plan for salvation than that. But this world is full of interesting people. Okay? But at any rate, so if you recycle this, you've still got to get the NAD back again. Then notice what you're looking at as far as evidence for creation. Anticipation. Okay, that's the big one. A lot of the steps only set up the next reaction. They accomplish nothing in and of themselves. They wouldn't have any survival value on their own. The complex prerequisites. Okay, you don't just make ATP out of nothing, you make it out of ADP. You just add a phosphate back again. And that's a double ring and a 5-carbon sugar and 2 phosphates already. There's a dozen of these protein enzymes that use, the average protein is 500 amino acids long. It uses 2 molecules of ATP to make each bond between amino acids, about 1,000 ATPs to make the average protein. And you've got lots of them in living cells. So you need lots of, and of course, DNA to make the protein. So you need the whole living cell in order to have an environment in which you can make energy to operate the whole living cell. Now notice the way God designs things. Once you get all these parts in place, what? You can make energy and have some left over to run the living cell. And you can continue to do it year after year, generation after generation, and so on. Just like if you designed a plant, designed a factory. They take a lot of energy to produce the first car, but once you got it all in motion, then you could just crank out cars, sell the cars, make the money to run the factory, and have lots of money left over, presumably. And so that's the idea here. You get all this evidence of complex prerequisites, looking ahead, anticipation as evidence of creation. Now at this point, Brian, do you want me to continue to talk now? I didn't know exactly how long that would take to say. I can say a few more things and then because we do want to have time for questions. But just to point it out, okay, we're looking, this is just a very simple metabolic diagram of what goes on inside your cell and mine. And all we looked at, let's see if I can even find it on there, is just this part right here. So that little blue arrow is all we looked at. And here's all the rest of the stuff. I thought about bringing the charts I used to study when I was in class, great big fold-out charts you make before the exams, and they are really impressive. Notice, though, at the end of fermentation, you've got molecules, in our case, lactic acid, with lots of hydrogen on it. And in aerobic respiration, using oxygen, those hydrogens are handed over to oxygen to form water. And that happens down here in this Krebs cycle in the electron transport system. We might take a look at those just much more briefly than I did the others. And so here, when you take a look at cellular respiration, some of you know, or most of you probably know, the overall formula is C6H12O6, a molecule of glucose, plus 6 molecules of oxygen, releases 6 molecules of water and 6 molecules of carbon dioxide. So when we aerobically burn sugar completely, it's a clean burning fuel. We take glucose and oxygen, turn it into water and carbon dioxide. That's all we get out of it. But all we talked about was this first step called glycolysis. Glucose is split into pyruvic acid. You get 2 molecules of ATP using this hydrogen conveyor belt here, NAD. What the Krebs cycle does, actually absorbing some water, is to strip all the rest of the hydrogens away from pyruvic acid and from water, hand them over to NADH2, and then the electron transport system hands the hydrogens over to oxygen to form water. There's a little energy made here, but over here there's 36 molecules of ATP made for 1 glucose molecule. We only get 2 anaerobically without oxygen, 36 more if we can use oxygen. And that's a phenomenal little process that it funnels. The hydrogens can be broken into electrons and protons. In the mitochondria, the powerhouse of the cell, runs these little proton pumps. As these protons flow through there, they twist enzymes that push ADP and phosphate together. And so it converts chemical into electrical energy, uses the electrical energy to push ADP and phosphate together. So that's pretty incredible. And in the overall process, where do you get the sugar from? And so ATP converts to ADP to energize life. Burning sugar takes the ADP, makes it back into ADP, produces carbon dioxide and water, and then it's green plants using light energy that take the CO2 and the water, put them together to make sugar. And so just briefly, very briefly, we'll take a look at that. And that's accomplished in these chloroplasts, the things that make green plants green. By the way, I guess I'll start here. This is a schematic. And so some of you have tried to fix a washer, dryer, a dishwasher, or something like that, and you look at the electrical schematic, don't you? And if you look at the little diagram, that they've got that shows how the wiring occurs, did you never notice how the wires always go straight from here to here, and then straight from here to there, and straight from here to there? So you look at the schematic and you say, yeah, that makes sense. And then you open the machine up, wires look like they go everywhere, So this is a brief condensation. The truth is actually even more fascinating than this condensed version here. But here you have sunlight. So here's the sunlight up here, and that's going to be the energy to put together carbon dioxide and water. That reaction occurs in 2 categories of